All right, yesterday we talked about macromolecules. So if you were absent yesterday or you missed that lesson, make sure you go back and watch the lesson first and then come back. The goal of this activity today is kind of unpack all the things we talked about yesterday with regards to macromolecules or biomolecules. And we're specifically going to focus on the structure of these biomolecules or macromolecules today. And I forgot to hit record, so I had already started this, but we're going to start with carbohydrates. And it says, what are the three types of carbohydrates? So yesterday we talked about the fact that sugars are included in our list of carbohydrates, and we have monosaccharides, which is just made up of one sugar, disaccharides, which are going to be comprised of two sugars, and then we have polysaccharides, which are many sugars. And so what we're going to do in our instructions, it says find the monomers of carbohydrates on page 2. So if you're doing the paper copy, this is page 2. Of course, if you're doing the digital version of this activity, it's a drag and drop, and you're going to build these digitally. So you might just want to kind of watch this video or watch the video and then head over and do the digital activity, or you can do it along with us. All right, we're going to color these carbohydrates, these monomers of a carbohydrate, red. And I don't have a red marker, so I'm going to use pink. So we're just looking at this, trying to figure out what our monomers are. And so hopefully you remember from yesterday that a monosaccharide is the monomer for a carbohydrate. So we're going to color all the monosaccharides on this page, which I believe is 5. We're going to color all of those red, or in my case, pink. And this doesn't have to be perfect. Of course, if you want to pause the video at any point in time or kind of fast forward through, you're welcome to do that. All right, so after we get these colored, we're going to cut them out and we're going to build them, glue them onto our paper, and we are modeling the process of dehydration synthesis, which, if you'll remember from yesterday's lesson, is the process of taking these monomers and combining them chemically to form a polymer. And this particular polymer that we're building now is a carbohydrate. So while we cut these out, let's review elements that comprise carbohydrates. Do you remember what those are? So it's going to be carbon, hydrogen, and oxygen. And remember, your carbohydrates include like breads and pastas and simple sugars like the sugar found in candy and fruit. And we talked about yesterday, carbohydrates are super essential for living organisms, because carbohydrates for me and you are going to provide our bodies or our cells with the capability to make energy. Now, if I'm a plant, carbohydrates are going to help in some structural support in the form of cellulose. But we cannot do without carbohydrates. Okay, so now what we're going to do, we have all of our monomers here, our monosaccharides, and we want to model dehydration synthesis. So we are going to bond these together to form a polymer. We call that polymer carbohydrates, but that's just made up of a long chain here of monosaccharides. So this is what we call a polysaccharide. This doesn't have to be perfect, just make sure that your little points here are touching. And that's representative of the bonds. Hopefully you did yours a little straighter than me. Mine's kind of crooked. All right, let's come in here and label this point here where the two monosaccharides come together. Let's just write chemical bond to kind of remind us that those are bonded chemically. Let's move on down to lipids. We're going to come back to this because it asks us for phospholipid to label the parts. We need to build it first though. All right, so there are different types of lipids. I want us to know or focus on the structure of a triglyceride and a phospholipid. So we're going to build both of these. Structurally, they're a little different, so we're going to model the building of those. And then we'll come back for our phospholipid and label the head and tail either hydrophilic or hydrophobic. All right, so it says for our triglyceride, we're going to come in with yellow. We're going to find a glycerol and 3 fatty acids, and we're going to color those yellow. All right, so I have a glycerol here. I'm going to color that. And then I'm going to find 3 of my fatty acids, color those yellow. And we're going to cut those out and paste them down. Alright, so just a reminder from yesterday's lesson, the elements that comprise lipids are going to be carbon, hydrogen, and oxygen, just like carbohydrates. Lipids are also similar to carbohydrates in that they provide energy, but the energy that lipids provide is long-term or a storage source of energy. Whereas carbohydrates are like a fast source of energy. Your cells will always use carbohydrates for energy first before tapping into other sources for energy. Fats also provide, or lipids also provide insulation. And then also we know, we talked about this yesterday, that they create phospholipids, which protects our cells. Those phospholipids form our cell membrane. Right, so our glycerol, for our triglyceride, we're going to turn it sideways. And then we're going to take our 3 fatty acid tails, and we're just going to extend those off of our glycerol. So if I show you something structurally that includes a glycerol and 3 fatty acid tails, you need to be able to tell me that that's a triglyceride. And now you can see, now that we've built it, you can see where we get the name triglyceride. Because the prefix tri means 3, and we have 3 fatty acids. All right, now we're going to build a phospholipid. So I'm going to need a phosphate group, which includes a phosphate and a glycerol together. And then this time I'm going to use 2 fatty acid tails, and it tells me to color those yellow. All right, so I've got my phosphate group here. Again, this is going to be a phosphate and a glycerol, which is one component of our phospholipid. And then we're going to have 2 fatty acid tails. So I'm going to have to do this sideways. So my phosphate group goes here. And then 2 fatty acid tails coming off the side. Right now I'm going to go back to the side here because it says label the parts of the phospholipid that are hydrophilic and hydrophobic. So just a reminder from yesterday, first, all lipids are hydrophobic. Fats, oils, waxes, steroids, hydrophobic. This means they hate water. So hydro means water, phobic means fear of. So these molecules are not going to like water. However, hydrophilic molecules are going to love water. So we're going to go to our phosphate group and we're going to label this hydrophilic. And again, we talked about this yesterday, the phosphate group, these are going to, excuse me, attract to water. These fatty acid tails are hydrophobic. They are going to repel water. And we know this because these are fats, fatty acids, so hydrophobic. All fats, all lipids are hydrophobic. All right, moving on along, flip over on the back. We've got proteins, and this question says, what are the elements found in proteins? So we've got carbon, we've got hydrogen, we've got oxygen, and we've got nitrogen. All right, now it says we're going to find the monomers of proteins. So just a reminder, the monomers of a protein, these are going to be our amino acids. So we're going to color all the amino acids blue. Cut those out, and then we'll link those together. We mentioned yesterday in the lesson that there are 20 common amino acids, and we're going to dig a little more into amino acids when we get to our genetics unit, specifically when we start talking about protein synthesis, because our cells will actually build proteins for us, for all living organisms. The cell will build proteins, and it does that through a process called protein synthesis. But in that process, we're going to have the linking of amino acids. And we're going to get real familiar with those 20 common amino acids. All right, at some point, I lost one of my amino acids. I think you're supposed to have 5. I only see 4 for me, so I'm just going to link the 4 that I have. If you can find all 5 of yours, link all 5 of yours. We're just going to link them together to represent that chemical bond that kind of holds them together like glue. When you link amino acids like this, we call this a polypeptide chain. And that polypeptide chain creates the polymer, which we know is a protein. All right, let's finish off with nucleic acids. Whoop, just ripped mine. All right, so what's the name for the monomer of a nucleic acid? It's going to be a nucleotide. Alright, so one of the things I want you to do as you color these nucleotides green, so it says find the monomers of nucleic acids, color them green. As you color these, I want you to make note that there are 3 parts to these nucleotides. So like here I'm coloring the phosphate group. You've also got a sugar. And there's also what's called a nitrogenous base. And we're going to learn when we get to genetics. There's some options we have for those nitrogenous bases. Alright, so let's color those green, cut those out. Now, in real life, the nucleus of the cell contains 3 billion nucleotides. So 3 billion of these. In this activity, we're only coloring 3, not 3 billion. But it's only to model the structure. So just to review nucleic acids, there are 2 types of nucleic acids. We have DNA, which is a double-stranded molecule. The 5-carbon sugar is deoxyribose. And then we have RNA, which is going to be single-stranded. And the sugar in RNA is going to be ribose. Alright, let me show you how we're going to link these together. So I'm going to put down this first one. I'm going to put it down sideways. Alright, when these nucleotides link, they're going to link at the sugar. Now, this process is a lot more complicated than what I'm sharing here, but since this is an introduction and we're talking about macromolecules, all you need to know is that they bond together right here at the sugar. And again, this is a chemical bond here. And we'll go into some more depth on this when we get into our genetics unit. Rip mine a little bit there. Just glue that together. Alright, and that is what a nucleic acid looks like. When you finish with this, make sure that you stick this in your notebook if you're doing the paper version. If you're doing the digital version, you'll need to make sure that you submit that.